Okay, let's get started. Hopefully I'll make it through today's lecture without succumbing to some single-stranded RNA virus, which has been so kindly brought to be by my daughters, because that's what they do. Viral vectors, that's their favorite thing to do. So today we're going to talk about transcription, which is why I didn't bring my actually really incorrect model. If people are interested in looking at that, kind of my office hours, tell me what's wrong with it. But of course, we have to have a couple of questions on repair, so we'll start out with those. In homologous recombination, Rec-A binds to single-stranded DNA with a 5' end, double-stranded DNA, single-stranded DNA with a 3' end, RAD51, or pCNA. $15, $10. Don't want to sound like NASA here. Countdown. Okay, so why can neither D nor E be correct? So both D and E are eukaryotic proteins. Rec-A is a bacterial protein. So I had to come up with some way of getting some of those answers. Fortunately, not very many people answered that. So brings us to single-stranded DNA with a 5' end, double-stranded DNA, single-stranded DNA with a 3' end. So what is the main thing that happens sort of first thing in homologous recombination? You have a double-stranded break. What happens after that double-stranded break? You chew in at one of the ends, which is which end? The 5' end gets chewed in. And so that leaves you with the 3' end, which is the piece that gets bound to. That's the strand which does strand invasion. So any questions about that? There seems to be some confusion about 5' and 3'. Maybe I should have brought the model with me. But unfortunately, the problem with the model is that I'm giving this away now so no one can get extra credit. But the way it's built actually has 5' ends right next to each other and 3' ends right next to each other. How the people who put it together managed to do that, I have absolutely no idea. Actually, it's pretty impressive that they were able to do that and actually get the model to fit together. So our next question is the mechanism of gene conversion, how it happens, is due to incorrect proofreading, base excision repair, nucleotide excision repair, non-homologous end joining, or mismatch repair. 20, 15, 10, okay, it looks like basically everyone's got this, so I don't need to go over it in too much more detail. The main thing here is that the mismatches come from the homologous chromosome. And so it wasn't any kind of DNA damage. It wasn't any kind of problem with proofreading. It was just because it happens that your two different homologous chromosomes are different relative to each other. So that's, that's the reasoning here. Again, it's not a DNA damage issue at all. Okay, so today we're going to switch gears considerably and let everybody leave. Maybe I should do clicker questions in the middle of lecture next time. No, shaking heads at the back for everybody who's leaving. Just as a quick aside, we are in the process now of trying to hire someone in the biology department whose research focus is going to be on teaching and teaching effectiveness. And one of the things that you may or may not have heard about, active learning, is supposed to be terribly wonderful for you. At least in some situations, it seems to make a change. Our seminar speaker, actually the candidate that came yesterday, talked about some flipped classrooms, how they had active learning and non-active learning, and looked at the differences between test scores before and after. And you know what the result was? Zero difference whatsoever. So interesting in terms of thinking about there. So maybe this whole lecturing thing is not so bad after all. So that being said, again, we're going to talk about transcription. This is just our overview that we've had before. So far, all that we've talked about has been this process, DNA making more DNA, replication, repair, and recombination. Now we start to move through the central dogma and talk about how we go to making RNA. Later we'll talk about protein. But for the next couple of lectures, we're really going to focus on this RNA synthesis step, also known, of course, as transcription. Exactly. What does transcription? It's our friend RNA polymerase, and I should actually be more specific here. This is the DNA-dependent RNA polymerase because it uses a DNA template. Again, that's what's happening here in our central dogma process. The real key to transcription is where you start. And so that process, also known as initiation, again, not that dissimilar from looking at replication, that's going to determine what genes get turned on and what genes get turned off, which RNAs are made at what times. And so it's really that initiation step which is absolutely critical for how you get which RNAs and that you get the appropriate RNAs at the appropriate times. Turns out there's some interesting differences between bacteria and eukarya. Curiously enough, not at the level of the polymerase, because the polymerase is actually pretty darn similar between bacteria and eukarya, but in terms of the way that it finds the right place to start. And one of those major things probably has to do with nucleosome-bound templates that you have in eukaryotic systems. We'll also finally talk about my favorite group of organisms here in a little bit more detail, the archaea. And it was, in fact, in the study of transcription that I got interested in archaea in the very first place. So why is transcription so critical? This is an image from your textbook. Oh, apologies to those of you who downloaded the PowerPoint between 9 and 9:45 p.m. last night because it had a different image in it. But this is a portion, a very small portion, of one of our chromosomes. That X chromosome, so those of us here like me who are genetically disadvantaged only have one of them. But basically, if you look at this tiny little piece of the chromosome, which is indicated up here, the X chromosome, which is in fact one of the larger chromosomes that we have, then look at that piece, just look at what's encoded in this particular piece. And so what you see are, to start with, a lot of areas that seem to have nothing encoded in them, these sort of gray areas. Nothing that we know of is obviously encoded there. And then you've got these horizontal bars, which are representing, I say vertical bars, I should say, which are representing exons. So these are coding for proteins. And one of the ways that you can tell that these guys are coding for proteins is just looking at this line down here, the blue. This is telling you the conservation between multiple different organisms' genomes. And what is really striking, hopefully is really striking, is every time you have a peak down here where these sequences are shared in a whole bunch of organisms, that's where you have protein coding sequence. And that's, in fact, one of the ways that people found protein coding sequences. You guys got this whole stretch of Cs, As, Ts, and Gs. How do you know what's actually being used to encode a protein? Since a lot of exons are pretty short, one of the ways to do that is to use this conservation process. So it's a very useful way of finding out what are coding sequences. On the other hand, you'll also see there are a couple of places, like over here, we've got pretty good conservation, but no obvious exon. So that could be a functional RNA, which is distinctly possible. The ones that we know about on here are actually relatively few and far between. There are these diamonds right here underneath. But there probably are a whole bunch more which are in there. We just don't know what they are, what they're doing. These are only the ones which have now been experimentally determined to be important. Another interesting aspect, and this is one of the reasons that I particularly pulled up this version of the figure rather than the previous one that I had, is that between the 5th and 6th edition of the textbook, the Neanderthal genome was finished, which is really pretty amazing. We can do comparative genomics on extant organisms, but also on extinct organisms, or at least relatively recently extinct organisms. And Neanderthals are really very, very closely related to us. And the indication of that is all of these yellow arrows down here represent where there are differences between the Neanderthal genome and the Homo sapiens genome. So it's pretty few and far between. And so people are concentrated in looking in some of these regions and say, okay, what's now the difference between Neanderthals and Homo sapiens, and getting some ideas about how that could be. So again, pretty few and far between. And then the other thing on here is that number of genetic diseases have been mapped to the X chromosome. And one of the reasons for that is, turns out it's a lot easier to map diseases to the X chromosome because, again, half of us only have one of them. And so you don't have two copies where you can actually have a gene very often on one copy, which is compensating for the lack on the other one. And so a lot of these areas here, which in fact we don't know exactly what the changes are. There's a change somewhere in this region here which is causing hemophilia. There are changes in this region here which are causing color blindness, et cetera. So it's a mapping of a region. Again, we don't know all of the details on that. So this, again, basically is my notes, which I've had on the side here. It's pretty hard to actually predict, just looking at sequence, where your genes are, what's actually being encoded. Certainly this homology, again, the blue line down here at the bottom helps a lot, but it's still pretty hard to predict. And there may well be some particularly non-protein coding genes that are in our genome that just we don't know just by staring at it. So yes, we've got the genome sequence, but understanding that genome sequence is a whole different story. And again, a lot of this has to do with the non-coding RNAs, which are present in the genome. So this could be, again, a whole course in and of itself if we just so happen to have a few more faculty who could be able to teach some of these things. So what do you make with transcription? You make RNA. Of course, it's ribose, uracil. One of the things that I wanted to emphasize here is that, yes, it's single-stranded. Well, I hate that term, but everybody uses it. RNA is single-stranded. I think a better way of thinking about it is it's one molecule. So it just has one 3' end and one 5' end. And so it's a single molecule, which then base pairs most of the time. And so that's a nice example down here at the bottom. This is what these RNAs really look like. That, to me, doesn't look single-stranded at all. That's clearly mostly double-stranded. One of the other interesting aspects about these base pairs, which form in our single molecule, not a single-stranded molecule, is that many of them are what people call non-standard base pairs. And I'm actually just looking for some of them in here. I should have tried to find some of them earlier. But there's a nice example right here, GG. So GG base pairs that form in RNA. So you've got lots and lots of different kinds of base pairs that are forming in RNA that you don't have in regular double-stranded DNA. And a lot of that is giving you this double-stranded or even the, in some cases, almost triple-stranded. Because if you look here down at the bottom, you can see here are a couple of bases which are actually sort of reaching over to another strand and making hydrogen bonds to that. So some of these base pairs actually are forming not base pairs but base triplets. Really fascinating kinds of structures that are going on here. And a lot of this is being done, the work trying to understand this in the chemistry department here at PSU. A lot of people are working on RNA biology and how these things fit together. So the standard base pair that you get, Watson-Crick base pairing, is, of course, adenine with uracil. And it's because we're missing the CH3 group here. That's the difference between uracil and thymidine, as well as the ribose versus deoxyribose. And the best way of actually describing this is not ribose versus deoxyribose. You should always be seeing 2' deoxyribose, because it's at the 2' position that you have a hydrogen here in deoxyribose and an OH here for your ribose molecule. We'll see that this OH is really, really important for splicing a little bit later on, but also because of this OH, RNA is a lot less stable than DNA. And anybody who's actually tried to work with RNA in the lab knows that this is a major problem. The other major problem is that there are tons of RNases all over the place, fingers, skin, you name it, and they're really hard to get rid of. So what kinds of RNA do we have? Most of the time people think about messenger RNAs. That's sort of the classic. It's part of the central dogma. DNA goes to RNA, goes to protein. All this RNA is in the middle is just this messenger that's not terribly useful for anything other than getting from the DNA to the protein. Turns out that's a relatively small amount of the RNA which is in the cell, depending on what cells, what kind of growth conditions. Anywhere from 3 to 5% of the RNA in the cell at any given time is messenger RNA. And this is partly why it took a long time to discover them, because they were kind of swamped by everything else which was present inside the cell. And we'll talk a lot more about that in molecular genetics next term. And if people are interested, by the way, thinking about what classes to take next term, I'll be teaching molecular genetics and virology and also the recombinant DNA techniques lab, so avoid all three of those. But you can also ask me for syllabi, and I can send them to you if you're interested in taking a look at that. So we'll talk a lot about why messenger RNAs were hard to find and how people, in fact, did find them and discover that that was the process that was being gone through. The main reason that they were hard to find was that most of the RNA in the cell is ribosomal RNA. And it's that ribosomal RNA which, of course, is important for translation later on, but this has all kinds of modified bases, bizarre base pairs, et cetera, and it's very stable RNA, which is present inside the cell. So you have lots and lots and lots of these. You have quite a few transfer RNAs, which are the little adapters, like called the little decoder ring for those of us who are old enough to remember Tracker Jack, which then will take the appropriate amino acid and attach it to the appropriate sequence, basically decoding the genetic code. You have quite a lot of these as well. Most of that rest of 15% are these tRNAs. But a lot of these guys down at the bottom here, again, those of you who have the 5th version of the textbook will notice there are quite a few changes here relative to that. And that's where really, as far as I'm concerned, molecular biology is moving really fast. Yeah? Would I like you to know for the exam, which I just added, these different kinds of RNA? I would definitely know the major ones and the ones that I talk about in class in more detail. But since I haven't written the exam yet, I don't know. But I consider it to be fair game. Unless it would be something like the, should I have them on here? Yeah, there's the small Cajal RNAs, so the CA RNAs that aren't on this list but were on last year's lecture or 2 years ago's lecture. That I wouldn't expect you to know. So, but this is expanding. This list is becoming ridiculously ginormous. And again, it's a lot of this non-coding RNA, which again could be a whole course in and of itself. But yeah, the main ones, mRNAs, ribosomal RNAs, tRNAs, snRNAs, and snORNAs. We're going to talk much more about these a little bit later on. And these are actually pretty well known, even last couple of versions of the textbook. The really new ones are these mostly regulatory RNAs, which are down here at the bottom. So microRNAs, small interfering RNAs, pRNAs, and long non-coding RNAs. So these, again, are the newer ones. We'll talk a lot more about them, again, as we move through the rest of the course. But most of these are regulatory RNAs. And a lot of them, we don't really know what many of them do, particularly these long non-coding RNAs. We know in a couple cases, like X inactivation, they're important for that, but in many other cases, that's not the case. So why bother with RNA? Why have this messenger intermediate between DNA and protein? Why not just go straight from DNA to protein? And people thought that's the way it happened. Probably the main reason to have a differential way of going from your genetic material to your actual protein is the possibility to get amplification. You can make many, many copies of one piece of your genome. So if you happen to need a whole bunch of one particular protein, say histones, one way to get a whole bunch of them is to make lots and lots of RNA from one particular region of the genome. Turns out for histones, even making a whole bunch of RNA is not enough. You need a bunch of copies of the genes. But that's another way of doing it. But as far as the idea here, the concept is that you can have amplification of one particular region of the DNA. And this can be differential amplification, different times, different cell types. Even if you've got similar genes in different organisms, they can be regulated in different ways. So this amount is really important. And then how you determine what that amount is going to be basically everything after the next midterm that we talk about. Some exceptions. Just don't forget, you can also get translational regulation as well as transcriptional regulation. But if you think about it from an energetic point of view, it makes much more sense to regulate at the beginning rather than trying to regulate at the end. Because if you've made all this RNA and you're not going to use it, it's kind of a waste of energy. So how do you make RNA? Again, this is the RNA polymerase. RNA polymerase conceptually functions very, very similar to the DNA polymerases that we've already talked about. Starts with double-stranded DNA. Those two strands come apart. And then one of those strands, the template strand, is copied. So you've got templates for DNA polymerase. You've got templates for RNA polymerase, and you make the complement of this template strand. Conveniently, of course, turns out it's exactly the same sequence as the non-template strand, just with the change of ribose and uracil instead of deoxyribose and thymidine. So if you want to know the sequence of your RNA, it's actually a lot easier to get the sequence of your complementary strand of DNA than it is to get the sequence of the RNA. Partly because it's so unstable, et cetera. So makes this complement. A couple of big differences relative to DNA polymerases. Yeah? So yeah, well, so the question is basic. I'll paraphrase it here. Is it always one strand that they're using? Or could they use both? And the answer is you can use both, and we'll talk about how that happens in just a second. So it turns out that that's one of the neat things about RNA polymerase. It depends on which direction you're going, which strand you end up using as a template versus a non-template. And that reminds me that polymerization works how? I think I heard it. 5' to 3', just like DNA polymerases. So always 5' to 3'. If there's nothing else you get out of this class, just 5' to 3', 5' to 3', 5' to 3', 5' to 3'. So that's the way that they elongate. Again, differences are that you don't need a primer. and that's partly why the DNA primase that starts replication is also an RNA polymerase. RNA polymerases don't need primers. They can start anywhere. So no need for primers, which of course means they need to figure out exactly where to go and start, and we'll talk about that in a little bit more detail. Just like DNA primase, they've got lower fidelity than the DNA polymerases. They can make mistakes, but since most of the time these RNAs are just used to make protein and then discarded, there's a lot of flexibility, A, in the genetic code, and B, these things are not maintained most of the time genetically. So you make a couple mistakes, it's not a big deal. And there are ways of dealing with that as well. There's a little bit of proofreading. In fact, people just found this out in the last couple of years, that if the wrong base gets incorporated, then it will get hydrolyzed, and then the polymerase will extend, the RNA polymerase will extend. It's not a separate, however, proofreading domain. This all happens at the active site in the polymerase, which is right down here in this cartoon model of what these guys look like. We'll see an actual higher resolution image in just a second here. But basically, you've got the double-stranded DNA here. The polymerase moves along in this direction, or the DNA goes through it in this direction, which is probably more likely. You have separation of the two strands. Unlike the case in DNA polymerization, we have a separate helicase. The RNA polymerase does it itself. So the RNA polymerase is actually the helicase activity, at least during elongation. Initiation is a slightly different story. So it's its own helicase, and then it makes the RNA basically right here, again, through base pairing with the template strand. And this RNA, the transcript, what's just being transcribed, comes out of the RNA polymerase complex relatively quickly and, again, relatively rapidly released. So this is the actual process. Down here, you can even see this happening. This is, I should say, these are a couple of ribosomal genes that are being transcribed. So the dark line in the bottom here is the DNA and all of the RNA polymerases that are sitting on it. And then all these little lines coming off of that are each of those RNAs. And at the end, it's probably the ribosome that's getting put together right there. So you can actually see transcription taking place in the electron microscope. So let's start out looking at bacterial transcription. The overall mechanism here, sorry.
On the last slide?
Yeah.
So is it just a coincidence that they bow out like this? Like do they start at one end and then they start adding on?
I'm not sure I quite understand. So is it?
RNA polymerases add on to the DNA at the exact same time, or do they start and then compromise where they just continue as they go?
Okay,
It seems like the new mRNA strands that are coming off
Mm-hmm.
longer at one end than they are at the other, like they're older.
Okay, yeah, so maybe this is a little confusing here, and I don't, okay, this is not a great image, but it's the one from your textbook, so which is why I'm using it. But the base pair, the new piece of RNA which is being made right here. So that's where you're adding the nucleotide to the 3' end of the RNA that you've already made. And so the 5' end is out here at this end. Oh, yeah, sorry. So here?
Yeah, like it seems like they were added on over here and then they slowly added on the other end, and that's why the mRNA in the long run end of the molecule.
Oh, so this is so, okay, sorry, maybe I should have explained this a little bit better. So what you're looking at here is the actively transcribing genes here. Again, they're not protein coding genes. And so everything which is down here in the middle, this is a RNA polymerase that's almost done, the dot here, whereas over here at this end is an RNA polymerase which just got started. And so well, what's happened is you have all of these RNA polymerases lined up right back to back. And so there's one right at the beginning which just got started, and then the ones which are behind it have made more in that process. Does that make more sense? So if you think about it, maybe one other way of looking at it is that these RNA polymerases are starting here and they're going that way. They're going off to the right. And so they're starting at the point of the Christmas tree. And that's, you've just had, and then you just, and you're going along. And as one moves along, the next one gets on and it gets started. The next one gets on and it gets started. That makes more sense? Good. Okay, thank you. Well, thanks for bringing it up. If it's confusing, it's probably not the only person who's confused by that. Okay, but I think it's really cool that you can see, again, transcription taking place there. So again, bacterial transcription, this is an overall idea of what's happening in terms of transcription. This cycle is basically exactly the same. The big differences have to do with the initiation, how you actually start, and what the proteins are involved in looking at that. So let's start out up. Let's see, where's the best place to start here? Okay, so here you have the RNA polymerase, again, the blue blob, that interacts with DNA, and it's got to find the right place to start. And so the right place to start, unlike your origin of replication that we had last time, is now the promoter. So the promoter is basically to transcription what an origin of replication is to DNA polymerization. So it's the place to start. And again, it's really important that you find the right place to start because these RNA polymerases can start anywhere. So how does it find the right place to start? In the case of bacteria, it's due to the presence of this purple thing here, which is called the sigma factor. Now, molecular biologists, again, don't have much of a sense of, humor maybe is not the right term, but they're not terribly creative when they talk about names. And so the factor here, it's something which you need to get transcription to work, and very often it's a protein. And almost always it's going to be a protein. So we'll be talking a lot about transcription factors for the next couple of lectures. So the sigma factor is a protein that's part of the bacterial RNA polymerase that's really critical for getting to start at the right place, i.e. at the promoter. So this sigma factor, together with the RNA polymerase, basically searches to find the right place to start. And it's through the specificity of the sigma factor which says, okay, RNA polymerase, this is the place where you want to be. Once the RNA polymerase finds the right place, i.e. the promoter, then you have activation of the helicase activity of the bacterial RNA polymerase. That forms what's called an open complex, open just because the two strands are separated relative to each other. Then what happens is actually really kind of interesting, and people just found out about this in the last couple of years, although maybe not that many last couple of years, because when I was in graduate school they were discovering this, so late Stone Ages. But here what they found is that all RNA polymerases, bacterial or eukaryotic, go through this process called abortive initiation. And basically what happens here is the RNA polymerase makes a couple of nucleotides and then sort of thinks about it again, to totally over-anthropomorphize here. Makes a couple of nucleotides and then throws them out, makes another couple, throws them out, makes another couple, throws them out. Now, why is that so important? Well, one of the other things that I didn't mention last time about RNA polymerases, unlike DNA polymerases, they're incredibly processive. Once they get started, they are going to go all the way to the end of the gene, and they're not going to fall off. And basically what that means is once you make the commitment, i.e. you are going to be transcribing the gene, you're going to be using a lot of nucleotide triphosphates in that process. So this seems to be basically a control mechanism whereby you only make that full-length RNA if you're really, really sure that you want to. Plus, it gives you another way that you can have regulation. So again, usually you want to have your regulation that happens as early as possible because that's the energetically best time to have that. So abortive initiation happens. In the book, they talk about scrunching because we actually know what the structure is of the polymerase. It kind of gets squished. and it's making these, I'll get to you in a sec. Squish when it's adding these extra nucleotides, but then it gets to a certain point after it's made enough nucleotides together, then it undergoes a big switch in terms of its structure, and then it's highly processive and will go for ages. Yeah?
The abortive sections, like the repeat, they do it on the same exact place, or is it like they do a view it and then move to a new section?
So yeah, great question. So the question is are they basically spinning their wheels at the same place, or they make a little piece, move on, make a little piece, and make a little piece, and move on? No, so they stay in exactly the same place. So that's really important if you think about genetic code and how you're making these RNAs. So yeah, they just sit at the same place. Make a couple of nucleotides, sit there, make a couple of nucleotides, et cetera. And that's also what they mentioned I think was a scrunching process where we know the changes in the structure of the polymerase as it's undergoing these processes. So it doesn't make a big change. Yeah?
What kind of control mechanisms would happen after abortive initiation that would keep it from proceeding?
Okay, we'll talk a little bit more about that as we move on. So the question is what are the regulatory mechanisms? That's all for like after the midterm. But excuse me, there are a number of, again, factors, and not my terminology here, which are what are called elongation factors. And so those elongation factors can be very important at this particular stage, is how much abortive initiation you have before you transition into the elongating state. And that's actually here, which is the elongation state. Once you move away from the promoter, and this is also called promoter clearance, you have a big conformational change that happens in the RNA polymerase, and that's basically grabbing onto your DNA. So it doesn't come off until, again, you get to the end of the gene that you're transcribing. This elongation happens until you get to a site of termination. We'll talk about termination in a couple of minutes. Basically, this is, forget where the terminology came from, oily stretch of road and a speed bump. It's a secondary structure that forms in the RNA, usually next to a whole stretch of uracil residues, which don't base pair very well. And so basically what that does is it pops the RNA out of the RNA polymerase complex. This is one kind of termination, but that's the main kind of termination. It's the easiest one to follow. So you have termination. First the RNA falls off, then the DNA falls off. You have reassociation of the sigma factor, which you lost over here, and you can go around the cycle again and again and again. And we know partly why you've got that kinked kind of structure that we saw last time, because people have actually imaged now RNA polymerases. That looks like the RNA, and these guys look like the DNA. And this is the blob at the bottom of them. Yeah?
Are there, you talked about the high specificity of the sigma factors
Yeah.
for specific promoter regions.
Mm-hmm.
Are there then different sigma factors for different promoter regions?
Okay, so the question is, are there different sigma factors for different promoter regions? You've either been reading ahead in the textbook or you know too much. Yes, there are. And that's, in fact, one of the really great ways that bacteria can pick and choose which promoters to use. They have different sigma factors. But we'll talk about that again after the midterm. So forget that for now. But we'll get back to it. Yeah? Yeah. So, yeah. Totally cool stuff. And again, that's what my PhD work is. Maybe I'll bring my PhD thesis to class one day. That'd be kind of fun. So I was actually looking at alternative sigma factors. But what do they bind to? It's a great lead into this next slide. So promoters. And these are those specific sequences in the DNA that the sigma factor, together with the RNA polymerase, are going to bind to. How do we know about these particular sequences? Well, one way is just to get your RNA polymerase together with the sigma factor and just look and see what sequences it actually binds to. And then you sequence that DNA and you compare it to other sequences that you know that these things bind to. And what you end up with is what's called a consensus sequence. And so this is a kind of a bizarre concept, so I wanted to go through it in a little bit more detail here. So the idea is here, you get lots and lots of sequences. Literally, I think in this case, about 300 sequences of promoters where your RNA polymerase, together with one particular sigma factor, in this case, bind to. And then you just look at the frequency of nucleotides at any given position, what that actual nucleotide is. So for instance here, at minus 35, and so maybe I should explain this. The minus 35 is relative to where transcription starts, where you get the first nucleotide is added. So it turns out this is away from that, and that gets back to your question about which strands are being used. So this is relative to that first position that's being made on the template strand, which one that gets chosen. So it has to be then what's also called upstream versus downstream. That's relative to where transcription starts and where it's actually moving. So here, 35 nucleotides away from where your transcription starts, you have almost always a thymidine, followed by almost always a guanine, followed by adenine, cytosine, and adenine. No, I do not expect you to remember this sequence. Then, if you move between 15 to 19 nucleotides, what is 15 to 19 nucleotides? Relative to, I guess I should have brought the model with me. That's about 2 full turns of your DNA helix. So it turns out that these sequences are on the same side of DNA relative to each other. And here, at the so-called minus 10 position, again, 10 nucleotides away from where you start, you have very high frequencies of thymidines, adenines, adenines. We'll see this again when we talk about eukaryotic transcriptional initiation a little bit later on. So these are the raw data that you see up here. And again, it's just looking at a whole bunch of sequences, just counting up at any given position, how frequently do you find one particular nucleotide. So one promoter could have TTC ACA, and another one could have TTG ACA, et cetera. And so lots of these, again, just add them up, look at them. And this is another way of presenting this down here at the bottom, where you see just frequency-wise, at minus 35, these 3 positions are almost always TTG, but you have some of the other ones. Minus 10, almost always TA, less frequently in the middle, and then almost always a T here at this position. So this is just a way of looking at multiple sequences simultaneously. So why is this so important? Well, it turns out that if you have a sequence which is almost identical to that consensus sequence, the RNA polymerase with that sigma factor binds to it really, really tightly. And if it binds to that really tightly, that means you're going to get a lot of transcription from that particular promoter. If you now have a sequence that has a lot of mismatches, it doesn't actually have this TTG-ACA sequence, then you have a lot less binding by the RNA polymerase, and therefore a lot less of your messenger RNA and then that gene, which you have at that point. So that's just one way of looking at regulation. It's just how similar is the promoter to the consensus sequence. If it's really similar, it's going to be a nice strong promoter. You get lots of transcripts from there. If it's not very similar, it's going to be a weak promoter. You don't get very many transcripts from there. Yeah, back. So the question is, are they major groove or minor groove? What do you think? Okay, you think that they're in the minor groove. Why? Okay, there's lots of T's and A's there. That is true. But can you get sequence information from the major groove, just what the base pairs are? 4' and prime. It's going to be in the major groove. So these are going to be all in the major groove. So the major groove of DNA is where you have the interaction of the sigma factor that's going to be that specificity factor that says, here, this is the place to bind. Yeah?
So does there not seem to be any kind of a consensus sequence in between -35 and
So the question is there a consensus sequence between the minus 35 and the minus 10? Here, this gap here. And the answer is no. There's no consensus sequence there whatsoever. It seems to be completely random. And so if you were to go across and look at all these sequences, what you'll find is the frequency of each nucleotide is absolutely identical across there. So the spacing, on the other hand, which is part of the figure that I cut off, is actually very well conserved, because if you think about it, that's because they're now on the same side of the helix. So you can bind, and it turns out there are 2 alpha helices from the sigma factor, which each bind in those 2 major groups in terms of where they're going to be. So because, oh, sorry, yeah?
How big are the sigma factors that are binding them? Roughly, how many nucleotides are they covering?
So they actually, those sigma factors, and so it's a sigma factor together with the RNA polymerase. But when it comes down and actually interacts with the DNA, we'll talk about how these experiments were done right at the end of class. But it turns out that those bind to most of each of those sequences. So this is part of the sigma factor that binds to -35, and this part of the sigma factor that binds to -10. And they pretty much bind to almost all of those nucleotide base pairs. Okay, so this finally gets to your question about which strands are being used. And so basically, it's going to depend on where those -10 and -35 sequences are relative to where you're going to be getting transcription to take place. And so if you think about where you're actually going to have position 1, so here's an example of position 1. If you now have those sequences, 10 nucleotides and 35 nucleotides upstream of that, that's where the polymerase is going to bind. It's going to transcribe in this direction. On the other hand, if your -10 and -35 sequences are over here, now the polymerase is going to bind and go in the opposite direction. And so you can actually transcribe from both strands. And it turns out this happens all the time. As you get transcription, you get genes that are being transcribed on both strands.
So that's how they can determine which strand was transcribed when they're trying to go back and look to find the RNA sequence? Because they can look for them in those promoter.
Yeah, so okay, the question is, how do you then know which of these are being transcribed? And the best way to do that is that you're exactly right, is to look for promoter sequences. Because those promoter sequences are not symmetrical. You know, if you look at them, if you try and let's back up now and look at our consensus sequence, TTGACA is not the same sequence going back in the opposite direction. So if we're going to look, again, this is just one strand. So from 5' to 3', it's TTGACA. But this would be TGTCAA. So it's clearly not the same sequence. It's not that same sequence in terms of where you're going to be with your RNA polymerase. So again, said that this is the case, that you have both strands which are being transcribed. We know this very well for the E. coli chromosome. We have both strands that are being transcribed. Very rarely do you have both strands being transcribed in the same place and coding a gene. And we'll talk about why that is a little bit later on. Of course, viruses do this all the time. But at least in cellular organisms, very rarely do you have genes that overlap on opposite strands relative to each other. And again, we'll talk about that much more later. But here, just in the E. coli chromosome, you have lots of promoters that can be on one strand. And again, -10, -35 in this particular case relative to gene D. Here, -10, -35 relative to gene C. And there seems to be no particular rhyme or reason as to which strands you use for your transcription. So let's look at the actual structure of these polymerases. The main thing that I wanted to concentrate on here is, well, A, we've got these amazing three-dimensional structures of RNA polymerases, and Roger Kornberg got the Nobel Prize a couple of years ago for this. But the main thing here is you've got our active site right here, where you're adding each of the nucleotides. It's right in the middle of your RNA polymerase, which doesn't have a hand-like structure. It's not like the DNA polymerases. It means it's not conserved, even though it's got very similar activity. It seems to be functioning. You've made your protein in a very different way. So they're not the same family, DNA polymerases and RNA polymerases, at least not the DNA-dependent RNA polymerases. The other thing to mention here is that you've got this looped structure, which is protein, that basically pokes its way in between the two strands and holds them apart. So that's sort of the helicase activity that, again, you have with the RNA polymerase itself. And that's what seems to be holding the polymerase on. It doesn't have a nice PCNA-like loop structure or a beta sliding clamp. It's that piece of the protein which is holding on to one of the two strands as you're doing this transcription. And we know lots of molecular details about this, and if you're interested in more about it, talk to Dr. Bartlett, because that's the focus of the research that's going on in his laboratory here at Portland State. So that's what that's what the polymerase looks like when it's extending. How do you get from this really nice, compact. You've got a piece of the protein wrapping around the DNA. How do you get that to fall off? Because you don't want to transcribe the whole genome. You just want to transcribe the gene that you're interested in. So the way that seems to work is either the presence of a termination factor that we're not going to talk about, or these what are called factor-independent terminators. So factor-independent terminators are secondary structures, I should say sequences that lead to secondary structures. When I talk about secondary structures in nucleotides, that's, again, base pairing interactions that happen. So secondary structures here are going to be basically stem loop structures. That secondary structure, which forms here, is due to sequences which are complementary to each other. So they'll bind, form this stable secondary structure, and then there's almost always a stretch of uracil residues that are added in this RNA. Why uracil? Well, just like AT base pairs, AU base pairs only have 2 hydrogen bonds that are binding them, so they're weaker. So it appears that forming this secondary structure together with the grease, i.e. the oil here, which are these weak base pairing interactions, that pops the RNA out of the polymerase structure. And if you look at the polymerase structure, we haven't looked at this in so much detail, it turns out there's kind of a pretty small hole that the new RNA comes out. And as soon as you get a secondary structure that forms, that hole's not big enough for that RNA. So basically it pulls the RNA off of the RNA polymerase. So that's how you get transcriptional termination. Termination in eukaryotic cells is actually, so this is what happens in bacteria. In eukaryotes, it's a lot less obvious what's going on there. So let's shift gears, talk about eukaryotes, just to make things even more complicated. They don't have one DNA-dependent RNA polymerase. They've got 3. They're very similar to each other in structure and also in sequence. So probably they've derived from one original sequence, which we think has to do with archaea, which we'll get to in just a second. So these 3 RNA polymerases, again, incredibly creative names, 1, 2, and 3. The one we're mostly going to be focusing on is RNA polymerase II, so I'll talk about that one last. RNA polymerase I makes most of, but not all, of the ribosomal RNA genes. And so that Christmas tree picture that we were looking at before, that was actually RNA polymerase I that was making all of these ribosomal RNAs. RNA polymerase III mostly makes tRNA genes, but also some of the ribosomal RNA genes as well. Turns out also some of the smaller RNAs. But the vast majority of transcription, at least for the interesting RNAs that we care about, is done by RNA polymerase II. All of the protein coding genes that we know of are transcribed by RNA polymerase II. Lots of the small RNAs as well. So this seems to be also the much more diverse of these polymerases. Polymerase I and polymerase III, they're going to bind to very similar genes, just keep cranking those out all the time, whereas RNA polymerase II needs to be able to get to all different parts of the genome to be able to bind to and transcribe from that. Yeah?
So in eukaryotes, you use Roman numerals to denote RNA, whereas in DNA, you use Greek.
Okay, so the question is, there's a bit on terminology. What is being used in terms of describing each of these? And so you're exactly right. I'm just going to repeat this, is that for the DNA polymerases, Greek letters are used, like alpha. When we've talked about alpha, we've talked about delta, we've talked about epsilon a little bit, but mostly alpha and delta. The RNA polymerases are just going to be 1, 2, and 3. Sometimes in the literature they're Arabic numerals, but pretty much here we're always going to have 1, 2, and 3 as Roman numerals. If you think about bacteria, there's only one polymerase in bacteria. So if you talk about RNA polymerase in bacteria, it's RNA polymerase.
Sure, but they say sigma factor and all those.
Right, but then you've got the factors, which are sigma, etc. And just to make things even more confusing, we'll move to the next table. So these are the factors, again, which are involved here. And so trying to keep track of them, again, bacteria, the RNA polymerase is made up of 5 basically different polypeptides, each of those Greek letters. So beta, and again, annoyingly beta prime, alpha, omega, and sigma. And sigma is not actually on this slide because it's usually not considered as part of the actual active polymerase. On the other hand, the eukaryotic polymerases, and it turns out 1, 2, and 3 are all very similar to each other here. They're all called RPB, which again, it's literature-wise. So in some cases, RNA polymerase 1 is RPA, and then RPC is RNA polymerase 3. RPB is what they name all of these different genes. Again, I didn't make these things up. And I'm not going to expect you to remember all of these different things unless I talk about them in a lot more detail. The important thing here is that you've got a lot more of these proteins in the eukaryotic RNA polymerases. True for 1, 2, and 3, but we're just looking at 2 here. Then you have in the bacterial RNA polymerase. But these sequences are clearly similar to each other. The structures, as we'll see in just a second, are basically identical to each other. Just the eukaryotes have more. But the structure in and of itself are very similar. So if the largest subunits, for instance, in bacteria are beta prime and beta, they're extremely similar to these sequences and structures, which are the 2 largest subunits you have in RNA polymerase II. Yeah, there's a question over here.
I was just going to clarify, so the structure similarity is between like the polymerase as a whole?
Yeah, we'll look at that. So the structural similarity is the polymerase as a whole, and that's 1 slide from now. We're almost there. Okay, the other column here, what's that? Strange thing over on the other side there. Archaea. It's the other major group of organisms, molecularly as different from bacteria as they are from eukarya. But curiously enough, if you look at their DNA-dependent RNA polymerases, they've got a whole bunch more subunits here. And actually, most of those subunits are pretty similar to the eukaryotic polymerases. And if you look at their structures, this is from a paper, oh wow, it's getting a couple of years ago now, from Katsu Murakami's lab, where he actually solved the structure of this RNA polymerase right here in the middle. And it's a RNA polymerase, in fact, from my favorite organism, Sulfolobus sulfotericus. But if you look here, particularly the bottom part, sort of what people also call the claw structure, that in Archaea is basically identical to the claw structure that you have in bacteria. But even more importantly, if you look at the overall structure of the eukaryotic RNA polymerase, is RNA polymerase II from yeast, these guys are practically identical to each other structurally. Turns out there's only 1 RNA polymerase in Archaea rather than 3. So some interesting questions about why that is. And people also think that this eukaryotic RNA polymerase probably evolved from something that was similar to an archaeal RNA polymerase, because if you go back and look at how all of these things are related to each other, this branching of the tree of life, the last universal common ancestor of all cellular organisms was down here. Then bacteria went off by themselves. Archaea and eukarya are basically together before they split off, and probably that's where one of these RNA polymerases came from. So let's look at RNA polymerase II here really briefly. Again, this is basically the same structure, flipped around 180 degrees relative to the last one. Structure, activity, extension, 5' ends, flaps that close down on the DNA, highly processive, exactly the same between bacterial, eukaryotic, and as far as we know, archaeal polymerases as well. The big difference is these guys don't have sigma factors. They have lots of different so-called general transcription factors. So these are basically serving the same role as sigma. Now, why have a whole bunch of different ones relative to 1 sigma. Probably a lot of that has to do with nucleosome-bound templates, and also that most eukaryotic genomes are a lot bigger than most bacterial genomes. So that's why you seem to need so many of these. And again, alphabet soup-wise, these are now called the transcription factors, TF. Which transcription factor do you have? Which polymerase does it work with? It's now TF2. So these are all the transcription factors which interact with RNA polymerase II. So TF2. And then we have a bunch of letters. TF2A, TF2B, TF2D, TF2E, TF2F, and TF2H. We're not going to talk too much about TF2A, but we're going to talk a lot about TF2B, D, E, F, and H, so I would remember what they are. What do? Basically, again, the same thing as what sigma factor does. They get the RNA polymerase to the right place. How do they do that? Well, just like you have in bacteria, you have promoters and you have consensus promoter sequences. Only here, the consensus promoter sequence is mostly pretty wimpy. It's called a TATA box. And TATA is because, again, molecular biologists are lazy. They just read the sequence, thymine, adenine, thymine, adenine. Okay, we'll call it a TATA box. So that's at about 25 nucleotides away from the start point of transcription, and again, in an upstream place. This TATA box binds to the incredibly creatively named TATA box binding protein, TBP. TBP is one protein which is part of a complex called TF2D. So TF2D, transcription factor 2D, lots of proteins all together. Maybe it's kind of an aside, but the reason they have all these funky names is because they separated all of these proteins on chromatography columns. Fraction A became TF2A, fraction B became TF2B, fraction C became TF2C, etc. So that's where those bizarre letters come from. They have basically nothing to do with their activity, it's just how they were purified in the first place. So TF2D has TBP as part of it, binds to the TATA box. Then you have TF2B. TF2B, again, was one of these fractions. Turns out that binds to the DNA as well at the TF2B recognition element. So BRE, that binds as well. These are the two DNA binding complexes that happen there. TF2A, it's not on this list. I wouldn't worry about it too much. You don't need it at all promoters. Whereas TF2D and TF2B, you do. Then you need to get the polymerase on here. And that process needs TF2F, E, and H. Most important of these is TF2H. Again, most of these are multi-protein complexes. TF2H is sort of the granddaddy of those, the one that has the most of those different subunits. It has two particular important activities. One of those is a helicase activity, and that helicase activity pulls the two strands apart so that you can get transcriptional initiation to take place. And it also has kinase activity. What do kinases do? They phosphorylate things. They add phosphates. Where those phosphates get added is to this strange little tail thing here hanging off of the RNA polymerase. It's also called the C-terminal domain. That's where you get all of this phosphorylation taking place. Turns out that's absolutely critical for getting out of the abortive initiation step. So I didn't talk about that before. It's one of the other ways you regulate abortive initiation, by phosphorylating this C-terminal domain tail. So this is just an overview of what I just talked about. TF2D contains TBP, but then a whole bunch of other proteins which are in there, also called the TAFs because they're the TBP-associated factors. TF2B just binds to this B recognition element. F helps the polymerase come along. E brings in TF2H, and TF2H has the helicase and kinase activities. We'll talk more about eukaryotic promoters on what day? Friday? My brain would work? Yes, we'll talk more about them on Friday.